project Our project is on the comparative genomics to discover novel relationships between sharks and humans in the context of thymus development. I'm Janvi.
And I'm Gautam.
In our presentation, we will be identifying the thymus, forming our hypothesis, our methodology, results, and introducing our future steps. In 2018, there was a study published on NCBI, which is a library database, and it suggested that the increase of cancer in humans as they age is due to the decline of their immune system, contrary to popular belief at the time that somatic mutations were the sole cause for age-related cancer development. In the paper, they identified that therapies that reverse T cell exhaustion or restoring T cell production could be possible avenues of treatment. The T cell is a type of white blood cell and a key component of the adaptive immune system of humans. They mature in the thymus, which is where they get their T designation. The thymus is an organ in the immune system that produces T cells. Within the discussion section of that paper, the authors predicted that for animals who don't experience a decline in T cell production or the decline in their thymus, they would be at a lower risk of dying from mutation diseases like cancer. One organism they mentioned in specific was the shark.
From this information, we decided to test if there was a correlation between the thymus of the sharks compared to that of humans, and how the difference in the production of the T cells could possibly shed some light on the secret of their longevity. We decided to use comparative genomics to try and find the differences in the thymus genomes and DNA sequences between humans and sharks to help cancer therapies.
Our main methodology of analyzing and comparing shark data is through comparative genomics. This is a field of biological research in which researchers compare the genome sequences of different organisms with the goal of uncovering similarities and differences between organisms. To gather our data, we reference several papers over specifically two databases from NCBI and Ensembl. We collected genome data relevant to the thymus from elephant sharks, humans, as well as dogs. The main reason we gathered data on dogs, in addition, was to have another control to compare the shark data to. Moreover, data concerning cancer in dogs was much more widely available compared to data on shark's thymus.
So first we identified the set of genes for the comparative genomic analysis. The table on the bottom right shows the set of genes identified from one of our references. Then we had to find a way to access these genes in our genomic pipelines to perform the analysis. First, we tried downloading the data from Ensembl and NCBI. However, we quickly learned that if we wanted to move fast and iterate upon this quickly, we would need a better solution. That's when we decided to directly use the REST APIs provided by Ensembl. Then we settled on using Google Colab because it allowed us to swap between R and Python to quickly get our results. Currently, we have made a phylogenetic tree and Venn diagram, and we are working on performing more analysis. If we are successful, we hope to summarize and publish the results.
These are our phylogenetic trees we used to create this. We used based on the Spark gene, which is one of the 57 genes we were looking into. A phylogenetic tree shows the evolutionary aspect of certain species and where they branched off, specifically pertaining to a gene that's being looked at. Our tree shows that the Spark gene is carried across elephant sharks, humans, as well as dogs. Through the tree, we can see that human and dark spark genes are more closely related to each other, while the spark gene of the shark diverged earlier. This may explain why sharks have a unique thymus and unique thymus genes.
In order to find which of the 57 thymus genes are shared between the three species, we used Ensembl APIs to get information about gene orthologs. We validated our results by checking the Ensembl webpage to see if the Venn diagram ortholog information are correct. What this Venn diagram basically does is, we're just trying to find the similarities and differences between sharks and humans. And below you can see on the bottom right hand corner, you can see that's an example of how we verified it. We went into Ensembl and directly saw if, for example, this is a spark gene, we directly saw if this was an ortholog or not. And we did this for all 57 genes. Some of the challenges we've had is obtaining high quality gene data for humans, dogs, and elephant sharks. This has been a challenge because relatively this field is new, so it's hard to obtain data. We're still in the data collecting phase, and right now we're trying to access Genomics data from GitHub.
In the near future, we hope to improve our methodology and data compatibility. We also hope to analyze the difference in mRNA levels in the shark thymus compared to the human and dog thymus. We hope it may give us more insight into the shark thymus. Pertaining to the future of our research, we're hoping the possibility of using drugs to give humans a genome similar to those in the shark thymus. In addition to other researchers, Dr. Laura Hale, in particular, received a 5-year grant award through the National Institute of Aging. It was part of a program project to determine mechanisms that control age-related thymic atrophy, which is basically the decline of the thymus, and identify therapies that can rejuvenate immune responses in older individuals. These findings define a unique feature of thymic involution and that linked immune aging um, and could have implications for developing immune boosting therapies in older individuals.
We would like to thank our principal investigator, Mr. Kanha, for his guidance and suggestions, and we'd also like to thank AHGRP and the All of Children Foundation for giving us this incredible opportunity. Thank you, AHGRP and Mr. Kanha, for your help in this project. We're very excited to continue this.